So welcome to the Wildlife Society's webinar series. This is also part of a trilateral webinar, which we'll introduce in a moment. My name is Kaylee Byers. I am the chair of the Wildlife Diseases Working Group. And next year, actually, I'll be going off into the sunset as past chair, and Dr. Kim Pepin's going to be coming on as chair. So it's very exciting to have her here today in that capacity as well. Just to say, I'm an assistant professor in the Faculty of Health Sciences up in Vancouver, Canada, and today I'm joining you from the traditional unceded and ancestral lands of the Musqueam, the Squamish, and the Tsleil-Waututh peoples. And really excited for you all to be here. I'm sure we've got folks joining from all over, which is really exciting. So again, thank you so much for taking the time to spend some of your day with us. So as I mentioned, this is part of the Wildlife Society seminar series, but we're also doing this as part of a trilateral webinar series with the Wildlife Disease Association and AAWV, the American Association of Wildlife Veterinarians. And I'm going to turn this over briefly to Dr. Cassandra Andrew to introduce what that is all about. Cass?
Yeah, thanks, Kaylee. Yeah, super excited to start the trilateral webinar series with this topic and with the Wildlife Society. So yeah, just a heads up for everybody that we'll be having hopefully around 5 or 6 more webinars scheduled throughout 2025 that are a part of this trilateral partnership. The upcoming webinar will take place on June 12th at 4 p.m. US time and 1 p.m. Pacific time. And it's going to be with Dr. Kevin Keel from UC Davis. The topic is human footprints, viral high-rays, breaking the cycle of emerging wildlife diseases. And so we'll make sure to get the registration link for you all out today. In case you want to join that. And upcoming ones, hopefully we'll also have some continuing education available for live participation with folks.
Thanks, Cass. Yeah, so we're really excited to participate in this series, and it's a great opportunity for all of us wildlife health nerds to come together. It's also a great opportunity to share our newish logo with the Wildlife Diseases Working Group, created by Henry Adams. And so I want to highlight that there too. If you don't already know about the Wildlife Disease Working Group, I just thought I'd introduce us briefly. Again, we are a working group at the Wildlife Society. We have, I think, about 380 members right now, which is really exciting, with folks all across wildlife health and toxicology. And our role is really to study and transfer information related to promoting wildlife health and prevention, research, surveillance, and the management of wildlife health. And so here you see, some of our main goals are to facilitate communication, cooperation, enhance knowledge and technical capabilities, increase public awareness, and promote undergraduate and graduate student interest in this space, and to support them at our conferences. If you're interested in becoming a part of the Wildlife Diseases Working Group, please do join us. It'd be great to have you here. And yeah, welcome to the conference, or welcome to our webinar. And I also want to highlight that Teglin Taylor is here as well. She's our Treasurer Secretary of the Wildlife Diseases Working Group and has been a really important part of putting this series together. So thank you, Teglin. And I want to highlight before we get going today too, that if you like avian influenza, we've got more of that content for you at this year's Wildlife Society 2025 in my hometown of Edmonton. Such an exciting location, the northern Alberta. So if you're coming, it'd be great for you to join us. We're going to have a symposium on avian influenza through a One Health lens. Cass will actually be speaking as part of that symposium. And then I'll be running a workshop. One of my hats is in health communication. So I'll be running a workshop on communicating complexity clearly, developing impactful messages, and countering misinformation. So if you're interested in some of those science communication and mis and disinformation skills, please do sign up. It would be great to see you there. Okay, but without further ado, we have 2 excellent talks today, Dr. Wendy Puryear and Dr. Kimberly Pepin. So Dr. Puryear is a scientist at the Cummings School of Veterinary Medicine at Tufts University, studying how viruses are maintained, spread, and evolve in wild animals. And today, Wendy will be talking about a broad picture overview of influenza and lessons learned from the Northwest Atlantic. And so after that, we'll have a talk by Dr. Kimberly Pepin. And Kimberly is a research biologist at the National Wildlife Research Center at the USDA, focused on developing analytical tools for assisting the evaluation and implementation of wildlife disease management. And today is going to be talking about surveillance of highly pathogenic avian influenza in hens and wild birds more broadly. And so we're really excited to have this overview of talks. The run of show will be about 15 to 20 minutes per talk with some time for questions. You can put your questions in the Q&A and we'll facilitate some of those. And then if we have some time left at the end as well, we'll answer some additional questions. But without further ado, I'd like to turn the floor over to Dr. Wendy Puryear to start us off.
All right, thank you. I will go ahead and share my screen.
Looks great.
All right, thank you everybody for joining us today. And as mentioned, I'm going to hopefully bring us on not what is too much of a whirlwind tour with what we have seen with influenza in the Northeast US over the last 3 years now since it's been here, and show you some of the lessons that we have learned from this area, and then bounce it over to Dr. Pepin for the broader national level view of what we've seen with influenza. So I'm a scientist at Tufts University at the Vet School, but I am part of the CEER network, which stands for the Centers of Excellence for Influenza Research and Response, quite a mouthful. But really it just means that it's a large collection of people who are really focused on trying to understand how influenza is moving in wildlife, and several different groups that participate in this related work looking at wildlife surveillance of influenza. I'm going to be focusing on the Atlantic flyway, but there are other people that are part of this network who have more details and expertise to give on some of those other flyways as well. And before I really dive into things, I want to start by acknowledging that this sort of work takes a massive village to be able to do. Although I get the honor of being able to present to you some of the insights that we've gained, this comes from collaborations with a huge amount of organizations and a number of different wildlife professionals that are very dedicated and skilled, ranging from the local to the state to the regional to the federal levels. So this is really a very collaborative effort from a lot of different folks. All right, so let's go ahead and start with just an upfront discussion of what is influenza A virus. Make sure that we're all on at least the same starting plane. So in here, a lot of different things that are tossed around in the media and various other sources that all fall under the umbrella of influenza A virus. One of which that really takes center stage these days is the one that is referred to as bird flu, or high pathogenicity avian influenza, HPAI, H5N1, all referring to the dominant outbreak that we've been seeing panzootic around the globe for the last several years. There's also, of course, seasonal flu for humans, and then we have heard over the years about swine flu. There is equine influenza, canine influenza. These are all influenza A viruses. So really it means that they're all part of the same family. And one of the features of influenza A virus is that there are two glycoproteins that are on the surface of the virus. There's a hemagglutinin, which is HA or H, and then neuraminidase, which is NA or N. We know of at least 16 hemagglutinins that exist and at least 9 neuraminases that exist. And the way that these come together is how we name the virus. So just to now put it up onto those ones that we just started talking about, bird flu, that's an H5N1, seasonal flu, one of which is H3N2, H1N1 for swine flu, H3N8 equine flu, and a similar related one, canine flu. But all to say that there are a lot of different forms of influenza. And one of the hallmarks of influenza is that it has a segmented genome, which means that it can co-infect an individual host, and all of those genes can swap around, making reassorted viruses that are whole new variants and lineages of the virus, which makes it incredibly tricky for us to try to stay on top of. Just talking about that hemagglutinin and that neuraminidase, those alone, there's at least 144 different versions of influenza A that circulate in the wild. So wild birds are the primary reservoir where we see all of this diversity of influenza, and you can see this whole range of different subtypes. So there are lots and lots of forms of influenza that circulate out there in the wild. Now, of those over 140 subtypes circulating in wild birds, they are primarily in waterfowl. So that is the primary wild reservoir for all of this variation of influenza. And in most cases, it is asymptomatic to those animals. Depending on the species, on the season, on the age class, you can have anywhere from 10 to 80% of a bird population that is infected with an influenza virus. And again, in a lot of cases, you don't see signs of disease. It's also the case that you rarely see most of these forms of influenza go into mammals. Primarily, it's something we observe in birds. You can, however, have occasional spillover into other species, and that's noted even from the previous slides about the equine and canine and human. You can get influenza that goes into mammalian species. Most of the time when that happens, it's considered a dead-end spillover. So you get an infection, it might go through that particular cohort, that particular group, but it doesn't go forward. So it's a dead-end event. Trying to understand what is going on with all of these different forms of influenza that circulate out there, really at the end of the day, that brings us to why we are so interested in looking into wildlife, because wildlife really have a lot of secrets to tell us about what is happening with influenza circulating in all of these different reservoirs. I'm just going to very briefly touch upon two of the things that we've been able to delve further into through the work that we do here in the Northeast. One of which is that birds are not the only ones that can serve as a wild reservoir, and that birds do indeed transmit very, very well. So they are incredibly great at transmitting the virus. So touching briefly into other species outside of avian wildlife, we see a lot of interactions between wild birds, particularly coastal wild birds, and marine mammals. So seals haul out, they spend a large portion of their life hauled out on dry land, and they are interacting very closely with a lot of those coastal birds that we know have all of this variation of influenza. So we've been able to work with, again, a large number of groups to go out to the healthy pupping grounds, out to the rookeries, and to test what appear to be healthy pups to see whether or not we detect any influenza. So we've been doing this for about 13 years now in the Gulf of Maine. At this point, we've sampled over 1,000 gray seal pups. And here showing you a picture from one of the rookeries, just showing you the density of birds butted right up against the density of seals. And what we've seen is that we very consistently find influenza virus that is circulating in these populations. So going all the way back 13 years, it ebbs and flows. We see this biennial fluctuation, but overall we get averaging out at around 9% where we detect viral RNA in the pups that appear to be healthy in most cases. Every now and then we'll have some that have some discharge, but it's not clearly correlated with them having virus. We're still detecting this virus out there in those populations. We also regularly detect antibodies in these populations against influenza. And we've been able to test those antibodies to see that they recognize a broad range of those subtypes that we also see in wild birds. So this would suggest to us that it's not just about the wild birds with the virus circulating. At least with the marine mammal population, we consistently see virus there as well. Now, in terms of a very nice demonstration on just how well those wild birds are at transmitting virus, however, we had this very interesting natural experiment that occurred here a couple of years ago that I like to refer to as the Great Gull Stowaway of 2023. So what happened is there was a food waste truck in a coastal part of Massachusetts, and the food waste truck closed up and was bringing into the dump to dispose of all that food waste. And when they got to the dump, they opened it up and realized that they had 5 or 6 dozen gulls came stumbling out of the back of the food waste truck. They were covered in food oil. They were not in particularly great shape. They probably thought that they had hit the jackpot, but were questioning those decisions perhaps just a little bit later on. So they ended up needing to be captured and rehabilitated, to be cleaned up and released back into the wild. So it's a huge undertaking, in particular by Cape Wildlife Clinic and New England Wildlife Center and Tufts Wildlife Center Clinic, of trying to capture these birds, to clean them up and rehabilitate them and to release them back out. We were able to test almost all of these birds for influenza between the time that they were captured off of the truck and released back out into the wild. Every single one of them tested positive for influenza. So though it's possible, it seems highly unlikely that all of those birds went into that truck with influenza. More likely one or a few had it, and spending a couple of hours together, sharing very densely populated feeding source, by the time they came back out, they all had influenza. So that just demonstrates very well at just how rapidly, under the right conditions, virus can spread through these populations. We were also able to test several of them for antibodies, because we were left with the question of whether they just got surface contaminated with it. And all of the ones we tested also had antibodies. So speaking of densely populated birds, that brings us to high-path influenza. When you have a high density of farmed poultry, that is where we have had the scenario where we have a change that happens in influenza A virus that has led it to become a highly pathogenic avian influenza, which really is just defined by its capacity to cause mortality in poultry. But ultimately, it comes from those viruses that were circulating in the wild reservoirs, spills over into poultry. In the case of the form of H5 that is circulating over the last several years, that has caused so much panzootic around the world, it originated in the mid-90s in densely farmed animals in Guangdong, China. So in 1996, there were domestic poultry where the virus evolved into high-path influenza, the H5 derivative that we now know of. It stayed there for about 10 years, stayed pretty localized, before it made its way back into waterfowl. There were, again, some localized outbreaks that occurred, but still relatively contained. Another 10 years or so later, those migratory birds started to make their way around the globe. We did have an outbreak that happened in North America at that time, which at the time seemed massive, but is comparably small compared to what we have since undergone. We got that outbreak under control pretty quickly, and we didn't see it in North America again until late 2021. The virus did continue to circulate in the Arctic and in Europe, but it wasn't back into North America until late 21. At the point that it did arrive back into North America, it very quickly disseminated across North America, across Canada, up into Alaska, down into South America, and really has continued to circulate at quite a fierce pace around the globe. So here I'm just showing you places where there are confirmed outbreaks of high-path influenza since that introduction in the late 21. And this has had a dramatic impact on domestic birds, but our focus for this discussion is on wild birds, which it really has just decimated many of those populations. So particularly colonially nesting water birds and seabirds, in some cases, there can be up to 100% mortality. Shorebirds and gulls, we've had complete colony collapses in many locations. Raptors, several of which had been on the upswing, have had declines in recovering populations. And then in particular, naive populations down in South America had tens of thousands of mortalities in wild birds when the virus hits those regions. Just showing you an example going from the higher latitudes all the way down to Antarctica. You have examples of large amounts of mortalities in Caspian terns in Michigan and pelicans in Peru, Adelie penguins and squat in Antarctica. And really this has made its way out to across all of North America and in South America, Galapagos, Antarctica, really everywhere except Australia has managed to, they need to share their secrets with us, but managed to escape. So that was showing you in relationship to that initial incursion that happened, that initial arrival of high-path influenza into North America. And that has been termed the A1, that first incursion coming on over. We have had multiple independent incursions that have happened. So even across the Atlantic, we know of 4 that have happened at least. And across the Pacific, we know of 2 that have happened. So those are those new forms of slightly different versions of the virus coming into North America. Once it arrives here, we were mentioning there's all that different variation of all those forms of influenza that circulate in the wild birds endemically. So once you have that high path come in, it has the opportunity to mix and match its genes and to create a huge array of reassortments that we have seen back into the wild bird populations. Showing you here just some of those subsets of the B's, the C's, and the D lineages, but it really just means reassortments from those high path viruses coming in. So we are focused in on trying to get as detailed an understanding as we can of what is happening kind of at the front door, at the gate, as things come across the Atlantic, really sampling along the Gulf of Maine and trying to understand with as much detail as possible what viruses are coming in and how are they reassorting and how are they moving from wildlife through wildlife, and then how do they move from that localized region back across North America and other parts of the globe. So what I'm showing you here is a map of just one example from one year of sampling effort. The darker colored squares are towns where we have samples that have come from. And this is really just highlighting that our data is very heavily coastal bias. So really everything I'm going to show you is through that lens, that we're looking very closely at the coastal population of wild animals. And then I also just wanted to highlight that gulls in particular, at least within our region, they have been the poster child for both incursions of those new forms coming in and reassortments that then happen locally with the endemic virus that's already there. So just one example of this, we had a die-off of gulls in Provincetown, Massachusetts in February 2023. And through sampling from dozens of gulls within a pretty small geographic area over a pretty short period of time, getting sequences off of all of those animals just from that one population, we detected 2 independent incursions of H5N1 in those gulls, another independent incursion of H5N5, another reassortment, but still a new one coming in from Eurasia, and then 2 completely different lineages of reassortments that happened that we detected within that population, and then some additional influenza that wasn't even H5. So all this diversity was represented just within one species, within one geographic region at one time point. So we have really wanted to have a broader idea of what we see in wild animals outside of specifically looking only at mortality events, but casting a really wide net so that we can try to capture anything that is also asymptomatic and perhaps going under the radar. And that's where all these partnerships have come in, of working with folks that are banding healthy animals and the wildlife clinics that are receiving animals being brought in, either because they're sick or because they're orphaned or because in some cases deceased, but not necessarily that it is a mortality event in all cases. And we're looking at wild birds. At this point, since H5 has arrived, so over the last 3 years, we've looked at over 13,000 wild birds, about 1,700 terrestrial mammals, and about 1,400 marine mammals. We've also been able to do about 100 necropsies on wildlife as part of that sampling. So again, these samples come from active captures of completely healthy animals, in some cases fecal collections, in some cases animals brought into the wildlife facilities, and in some cases carcasses that are brought in by partnering organizations or field work that we also are doing. So ranges really from healthy to sick to deceased. We're collecting swabs on those animals. We recover the viral RNA, and then that is screened to look specifically for influenza A virus, broadly speaking, and anything that is influenza A, then additional testing to see is it H5. So a broad sweep overview of what we've seen, again, within this coastally biased sampling that we do, is that there really has been an expansion of these species of animals, of wildlife that are impacted by influenza since this form of HPAI has really expanded. Primarily what we see a lot of so these are all the samples that we have that have been H5 positive. So a lot of them fall into the category of seabirds, shorebirds, and sea ducks, which perhaps is not much of a surprise given the region that we're in, but that is where our dominant positive samples come from. We also see a lot of species that are of raptors. So falcons, hawks, eagles, owls, vultures. And then we also see ducks, which is to be expected with influenza, but we see it in ducks, geese, and swan, and then corvids. So we picked it up in both ravens and crows. But again, the bulk of what we see are in those seabirds, shorebirds, seabirds, and raptors. So this perhaps is not surprising, given that a lot of those populations, particularly with the seabirds, shorebirds, are densely populated species where, as we saw with the gull stowaway in the food truck, that you have that dense population of animals, it allows the virus to go through very quickly. Also, with all of the raptors, that represents some of the transmission that we have also seen become more common of a rule with scavenging and predation. Focusing here, so that feeds into the raptors, but we have also detected influenza in our area in both fox and seals. Oops, wrong way there. So this is just giving you greater detail on the 49 different avian species that we've seen just within New England, breaking down with a little more granularity for those big categories I just showed you. And globally speaking, there's been well over 500 avian species that have been detected with high path influenza. We've also been able to see that now that we've had 4 seasons of this, we seem to be settling into a bit of a winter pulse. So that first season, it was kind of all over the place when we were detecting high path influenza. That second year, it was still spread throughout the whole year, but starting to tightening into a January, February, March timeline. And the last 2 years, it's really been focused in kind of January, February, March, where we see the real spikes of high path influenza within our region. And I'm also superimposing on there the fact that each year with those winter pulses, we've also seen a shift in the dominant lineage that we pick up within our region. So that first year, the dominant lineage was an incursion that had come in, an A2 lineage. That second year, that was largely gone, and we had mostly reassortments. The third year, we had yet a different reassortment. And this past year, we're not sure what the subtype is yet. But the ones that we've detected so far have been H5N5. So we've been seeing a shift from year to year. So where does that leave us? Really, that further knowledge of those additional influenza variants continues to be critical. There is a large pool of influenza variants that continue to circulate out there, and they continue to feed into the reassortments that come in with those new forms of high path influenza, and provides ongoing and continued opportunity for the virus to evolve and to move into additional species and to shift the way that it is presenting clinically. It's all things that we need to understand the whole breadth of what is there for influenza. Species other than waterfowl, such as seals, may also serve as reservoirs for also this low path virus, these non-H5 forms. And those are something that we should continue to pay additional attention to. And under the right conditions, influenza transmits incredibly efficiently. There are some silver linings, possibly settling into a bit of a seasonality with wildlife at least. So perhaps it's not as chaotic and crazy moving forward, we can hope. There have been a handful of animals that have been able to recover in rehab, and we have certainly picked up animals that have antibodies against high path influenza, suggesting, of course, that they had been exposed and they did indeed recover. And that has been true in both birds and seals. So there's a hopeful edge to that. And then really just to leave on the fact that the dynamics of influenza A continue to be rapidly changing. High path continues to have new incursions that are coming in, continues to reassort, continues to change, and we continue to see this expansion into additional species, both wild and domestic. So again, I just want to circle back around to thank all of the collaborators and contributors who are absolutely instrumental to being able to collect this data and to try to get a better understanding of what is happening in wildlife and how all of those pieces come together to try to address some of these questions of understanding that disease ecology. If there's time, I'll take a few questions, but otherwise I can wait until after Dr. Pepin goes.
Thanks. Thanks, Wendy. That was so great. I know it's weird in a webinar space, so I'll do one of these to simulate everybody clapping, I'm sure, from their offices, either at home or at work. I'm going to ask you one of these questions, and then if you'd like, you can also respond in the chat to any of the other ones that we have, and then we'll go on to Kim.
Okay,
So one question that came up for a couple folks was around passerine birds. You know, they're small, they're hard to collect, they're gone on the landscape. Could there be more HPAI in passerines that's just not being detected? And could it be contributing to the decline of any of these species?
Yes, that is a great question, and it's a complex question. And I think Dr. Pepin is going to get into that a little bit with paradomestics. So I'll just give a brief glaze overview. It can be and has been detected in passerines. So we have had a few cases within our data set, not a lot, but we've had grackle, we've had a few cases, literally like maybe 5. It's also the case that these are animals that are easier to go unnoticed because they're small. They are not necessarily going to be observed and brought into a wildlife facility to be tested. So they certainly are also under tested. But we haven't seen that be a large area where there is high path influenza, with the exception of animals that are proximal to infected agricultural settings. And I'll leave that for Dr. Pepin to go into in more detail.
Great. Thank you so much, Wendy. And thank you everyone for your questions in the chat. We'll try to get to them via typing, and then maybe some of them will get answered in Kim's talk. And if we have some time at the end, we'll come back to them too. So thank you again, Wendy, for that excellent presentation. I learned a lot. And Kim, over to you to tell us a little bit more and get at that domestic to wildlife interface as well.
Yeah, thanks a lot, Wendy, for giving that comprehensive overview of influenza A in wildlife. I'm going to focus in on HPAI, in particular, the current outbreak of H5N1. And I'm really here presenting some slides for the National Wildlife Disease Program, which is part of USDA APHIS. And that's why you'll see Sarah Bevins is listed here as the author of this presentation, because most of these slides come from her, but she couldn't make it today. So the National Wildlife Disease Program, which is run out of Wildlife Services and USDA APHIS, is a program that's coordinated around the country with wildlife biologists in different states around the country. And there's a variety of priorities in the program. So the top priority being emergency response. So these wildlife biologists provide support for producers when they're experiencing an outbreak, and also during the current outbreak began doing peri-domestic surveillance around premises. And then another big part of the National Wildlife Disease Program is doing surveillance in apparently healthy birds, and also morbidity mortality events in migratory birds, and then some special projects, other disease projects. So I'm going to talk about 3 methods of surveillance run through NWDP today, one being their targeted surveillance of dabbling and diving ducks, and Wendy spoke a bit about this as well, and the morbidity and mortality investigations, and then also the peri-domestic surveillance, which occurs around infected premises. And the NWDP reports all of these data in public venues, such as these websites listed here, almost in real time. So there's all of the avian influenza and wild birds and mammals that you can access publicly. In terms of the routine targeted wild bird surveillance, the sampling seasons are broken up to sort of match the breeding cycles of wild birds. So the sampling year starts in May, and the summer season is May to August, and then the fall season, September to December, and then winter and spring, January to April. So the sampling quotas for the year are broken up along with the bird life cycle. And then sampling is across the 4 flyways with all the disease biologists across the country in 49 states targets are made in 49 states, and then there's opportunistic sampling in Hawaii and Puerto Rico and the US Virgin Islands. And each year there are annual target numbers that are developed based on the previous year's surveillance data and a bunch of covariate data. And this system has been developed by our colleague Ryan Miller, who's over at Veterinary Services and SIA. And last year, for example, the overall sampling targets for the season were 38,000, a little over 38,000 samples, and this year's targets are a little over 33,000. And just to give you an idea of how the sampling quotas are broken up across the years for seasonal targets. So this is really, the targets are designed to match where the birds tend to be at different points of the year, and where we're previous year's data suggests they're most likely to find avian influenza. And this just gives you an idea. You can see those 2 points there are where the first introductions of the current high path occurred in the US in blue-winged teal and American widgeon. And this video just sort of marches you through where detections have been happening over time throughout the current outbreak since fall 2021. And it overlays where, you know, detections in dairy facilities and poultry, and then wild birds sampled by the Wild Bird Surveillance Program, part of NWDP, and then from other agencies, the morbidity and mortality events. And you can see as this video progresses, down below here, you can see there's some seasonality in the detections, and you know, that's when all of the points are going brighter and not so bright, and that it's really all over the country these detections are occurring. And you can see some clusters in the dairy and poultry throughout. And this just sort of shows you the data. The black line are showing you the numbers of detections of HPAI, and then the blue bars are showing you the number of samples tested. And you can see that these things track each other fairly well, and that's partly due to the adaptive surveillance that Ryan developed in collaboration with Sarah and NWDP. And so the surveillance plan, to some extent, also provides a risk assessment. And then this kind of shows you the seasonal dynamics of that. So in the first year of the outbreak, there was a large number of detections happening in the spring, and then the detections in the fall started happening much earlier. But in later years, the peaks for across the country, Wendy showed some sort of later peaks of seasonality, but across the country, the peaks are starting in November. And then this very colorful rainbow plot shows you, the point of this is really to show you that there has been a lot of genotypic variation over time, with different genotypes coming in as being predominant and then being beat out by other genotypes. And you can see when B313 emerged in cattle down here, and that was not detected in wild birds, but now, more recently, there's been D11 is the predominant genotype, and that has also been present in cattle. And this just gives you a spatial view of the information with a lot of different kinds of genotypes all over the country from September to December, and more recently, that sweep of D11. And then in terms of the morbidity and mortality investigations, this is surveillance of sick or dead birds on the landscape, typically with neurologic signs. And these are sampled only when necessary and tend to be limited to a few individuals per event. And in terms of which species are sampled most often for those events, you know, it's ducks and geese and swans, and then raptors and vultures. And these samples are collected by a variety of partner state wildlife agencies, the wildlife services, disease biologists, and then rehab facilities and universities, and they make up about half of the detections in the broader data set. And then in just a bit of background on the H5N1 detection in dairy cattle, initially that was the B313 genotype, initially detected in a dairy premise in Texas in March 2024. And that initial spillover from wild birds was between October 2023 to January 2024. There were similar viruses to B313, but that exact virus wasn't picked up on the wild bird surveillance. And then it continues to spread throughout dairy and poultry facilities, mostly from anthropogenic movement of the virus is suspected in all cases, and with spillover to peri-domestic mammals and birds. And there have been multiple cases in farm workers and domestic cats, but no wild waterfowl detections. And then more recently, there have been 2 other introduction events of D11, and that's a reassortant virus derived from the A3 genotype. That was first detected in wild birds in September 2024, and then quickly spread to all the US flyway, as I was mentioning earlier. And it was first detected in poultry in October 2024, and then in dairy cattle in February 2025 in Nevada and Arizona. So this gives you the picture. There's still lots happening in high path AI evolution and spillover. And so as part of that, to try to better understand risk to producers, NWDP began deploying to premises to sample peri-domestic wildlife. And I'm going to talk about some of the data that were collected on 62 positive farms. 58% of those were dairies and 42% poultry in 16 states. So far, more than 165 different wildlife species have been sampled, and there have been detections of B313 and D11. And the surveillance design is really aimed at getting high path detections to explore which species might be susceptible. And so the design's opportunistic in terms of just trying to sample whatever species can be found, with the guidance of sampling for 2 weeks or up to 250 samples. And you can see here that the overall apparent prevalence so far is about 4.1%, with over 9,500 samples collected and 400 detections. And genome sequencing suggests so far that the spillover is mainly from domestic animals to wild animals. And as I was mentioning, both B313 and D11 have been detected. And so the answers so far from the data are that are wildlife bringing B313 onto livestock and poultry premises? It looks like the answer is no, but B313 is spilling over into wildlife from the facilities. And then is the virus moving off farm and persisting in wildlife? The answer appears to be no for B313, but it's unclear yet for D11. And so we want to dive a little deeper into the data, and this is still very much ongoing. So here's just a few preliminary thoughts about it. In terms of what are the highest risk species for informing ongoing surveillance? So now that we've done a lot of completely opportunistic surveillance, can we start to refine the sampling design to maximize our detections? And you can think about, there's several different factors at play here in terms of what species you can find on different premises and how widespread you can find them. And so we looked at these kinds of criteria for ranking species in terms of number of premises sampled, number of positive premises, average sample size across sites, and number of positives across all sites. And looking at that, the species that tended to be most infected were house sparrows, house mice, rock pigeon, European starling, Eurasian collared dove. Those were kind of in the top 5. And this gives you an idea of the average sample sizes that we're finding on the premises. So, and also these were sampled at between 27 and 45 premises. So they're very widespread. They're found widespread on the premises. And between 1 and 8 positive premises tested positive for these. And then you can see here that if you break the data out by the number sample size on all premises versus sample size on when you're finding them positive, you can see that there's a higher sample size when you're finding them positive, highlighting the importance of sampling enough to get a detection. And so it seems that from the data, the ones that are coming up positive have between 27 and 40 samples. And then this just gives you an idea of, so for these top 5 that tend to be positive, they are coming up positive on dairies and poultry premises with different amounts. So for example, house sparrows are positive on both dairies and poultry premises, whereas house mice tend to be only positive on the poultry premises, and rock pigeons tend to be positive only on the dairy premises. So there's some species level differences on premises that could be used to optimize the sampling design. And just to give you an idea, there's differences in prevalence of these different species. And when you compare that, so this is in poultry, so what you're looking at is like the sample prevalence across different species for poultry premises. And you can see there's a lot of variation across these species. These are ones that tended to be positive. And then when you compare that to the dairies, there's different species that tend to be positive, and the overall sample prevalence tends to be lower on dairies. And then you can turn around some of what we learned about the sample size to look at species that we tend to find and are most sampled, but we're not getting detections, and so maybe lower risk for infection. But one thing to keep in mind is when you look at these species that we're not getting detections in, they also tend to have lower sample sizes. And so we can use this information to better plan our sample sizes. And again, even with the ones that, so we can also use this information to help us understand where we're most likely to find certain species on dairy versus poultry premises. So when we're doing the sampling opportunistically, we tend to find different species on different premises. And so that's another more information for risk assessment. And last year at TWS, we did a workshop on just we developed this application to help better understand what sample sizes you might need in wildlife populations to have a certain level of confidence for detecting the pathogen you're interested in. And so we can use this application to ask that question to help us refine our sampling design so that we maximize detection efficiently. And looking at this, basically what we find is that a sample size of 80 would give you a 95% detection probability. And so in terms of just some preliminary results for what tends to be risk factors on premises, in terms of like sample prevalence, B313 tends to have higher prevalence than not B313, and poultry premises tend to have higher prevalence than cattle premises. And the higher the sample diversity, the lower the prevalence, and that is likely linked to a sampling size issue. And then also just in terms of a general trend, there's been an increase over time in sampling prevalence since the start of the peri-domestic surveillance from April 2024 to March 2025, and also an increase over time in detection probability. And in terms of some, like just wrapping it up on the sampling recommendations, so as I was mentioning, the model suggests that 80 samples could give you a 95% confidence of detection. And if we look at like house mouse, for example, where we found a lot of positives, we tend to have we tend to be sampling about 25 individuals, which means our detection probability should be, our confidence of detection should be around 56%. So we can use this kind of information from this application to refine our sampling design. And that's just, and some general guidance is that, we can focus on species focus on species that are most likely to be sampled and infected, and then also use that sample size app for guidance. And here's a nice picture of, as Wendy pointed out, it takes a lot of people to do all this good work. And so here's a picture of that from our annual meeting of NWDP wildlife biologists who do immense amount of work every day for this stuff. So yeah, we're all very thankful.
Yay. Thanks, Kim. That was great. It was really, yeah, it was a really great, you folks coordinated your talks really well to build off each other. I think what I'll do is ask you one or two questions, and then Wendy, maybe we'll have time for you both to answer one or two questions before we wrap up. So I want to sort of put two questions together. One was about sampling animals in and around premises, and you mentioned, you should be thinking about what are the animals that are most likely to be affected. So when you're thinking about doing that work, like I imagine there's multiple different sampling methodologies depending on what animals you're looking to gather. Can you speak a little bit about that?
Yeah, so I mean, the biggest things are kind of practical, like you're going to sample what you find, and then there's permitting that also limits sample number. So there are certain certain species that you can't sample in high numbers. So you want to kind of balance all of that if your sampling objective is to get a detection, if that makes sense. I'm not sure if that answers your question comprehensively enough, but
yeah, I'm just thinking like depending on like you're out there, my bread and butter is rats, right? You're out there sampling for rats, you're using a very different methodology than if you're going out here sampling for passerines in meat. Although I used to get passerines in my rat traps fairly frequently, though, if I'm being honest with you. Thank you for that. And then a question. Oh, go ahead.
I was just going to say, I can't speak for the on the ground sampling because I have not done it. All these wonderful people I just showed you are the experts.
And so obviously like testing all these animals is like challenging. You got to get out there, you got to grab them. What do you think about the potential for environmental DNA for doing some of this sampling in and around the sea?
Yeah, that's a very interesting question. I tend to think that is could be a useful direction. We've talked about it a lot in terms of, we know there's a lot of virus there, right? And so it really depends on your question. Like when you have a large poultry facility that is infected and it has a lot of virus, like sampling the environment around there isn't providing additional information. I think it would be more useful if you're wondering, like, is it present at all in the population, Whereas like when you have a bunch of virus around, like that's not going to tell you which species are most likely to get it, and is it maybe you could use it for a persistence question, I guess, like after the poultry facility has been depopulated. That might be a more efficient way to look at persistence, but
yeah,
it depends on your question,
Yeah, and I know we have a group up here at BC that's been working on eDNA sampling for HPAI, and there's all the lab challenges as well, right, about finding the best ways to go about sampling HPAI from mud. So it's also comes with its own challenges.
Yes.
I'll turn a question over to, oh, go ahead.
I was going to say there are several investigators working on that through the SEER network as well. And to both of your points, it's logistically very challenging. So it's a problem that is still trying to be solved. There is progress being made, but it's not quite there yet.
I have a question here from Veronica, and I'm going to take it and I'm going to shift it a little bit. So Veronica was curious about, the evolution of these viruses in the environment and whether that would help us look at possible future emergence of these strains. And then I also think about the wildlife angle in terms of there's a lot of wildlife out there and a lot is done through passive surveillance. So what do you think about studying the evolution of these viruses for that purpose? And what gaps do you think there are in effective wildlife surveillance and response?
You want
Oh,
Take that one,
Kaylee,
Wendy?
can I have another hour?
to? Right.
Yeah, can you please solve this?
How to address that quickly. So there are so many gaps. Surveillance is woefully underfunded, under-resourced, under-personneled. There are a lot of places that we just simply don't know because we don't have the resources to look. And in particular, trying to do that broad net surveillance is one thing, and we get a lot of really great information from looking at mortality events, but that still leaves us with that huge gap in knowledge about what is going on undetected that provides the raw material for reassortment to happen and to give the virus the capacity to expand into additional species because it has some new genes that those species haven't seen before. So it's a huge blind spot that we have that we are desperately trying to fill through all of these partnerships, but we need more resources. And as far as how the virus evolves, that is a question that is also incredibly important and all feeds into needing to have more of those viruses to be able to query, to be able to really characterize how those viruses are evolving and how that impacts the ways that they function. Just one quick part that I'll touch upon is environmental persistence. There's kind of a third component to transmission between host A and host B. There is an outside of the host time of transmission cycle. And if the virus is only able to survive for half an hour, that's very different than if the virus can survive all winter long and still be there come the next spring. And we do see that range of characteristics of how the virus can act, depending on some of those evolutionary changes that can happen in the virus. So those are things that we want to try to understand better as well, which help to inform those questions. And that took several minutes, so I'll stop there.
No, that was great. And I think it's going to be a good question to end on. Kim, is there anything else you wanted to add to that before I wrap us up?
No, I thought Wendy covered it perfectly.
Absolutely wonderfully. Okay, everyone. We have so many great questions here in the chat. I'm sorry we weren't able to get to all of them. Thank you for sending them in. I hope you enjoyed today's webinar. Thank you so much, Kim and Wendy, for taking the time to share your research and expertise and collaborations. Again, this work happens in very large teams. So a silent round of applause for both of you and all this work. Thank you
Kaylee,
again.
if I could just take one quick moment. There's a lot of questions we couldn't answer. I'd like just to extend an invitation if anybody wants to reach out by email. I will do my best to answer as quickly as I can, though that might mean it might take me a week to get back to you, but I will try my best to respond as fast as I can, and I am happy to continue these conversations.
Yep, same for me. I was going to say the same thing.
I think a week is a very reasonable timeline. Let's go back to handwritten mail, Andy, in the post. Thank you again, everyone, for coming. If you haven't done enough HPAI, we are going to do another avian influenza symposium at the Wildlife Society in Edmonton this year. And we also have a workshop on health communications through the Wildlife Diseases Working Group. Hope to see you at the next webinar and have a great rest of your day and end of the week. Thanks for coming by.